Welcome back and welcome to a new video on a group of fungi called Aspergillus, Aspergillus niger, Aspergillus flavus, Aspergillus fumigatus. These are the very important fungi under the genus Aspergillus. This genus Aspergillus was first documented by Antonio Micheli in 1729. The word Aspergillus is derived from a Latin word which means holy sprinkler. The holy sprinkler which is used in churches. The genus Aspergillus has about 339 species distributed worldwide. Some of them are highly pathogenic and they cause a number of diseases. One common disease caused by Aspergillus genus is Aspergillosis, which is a lung infection. People who were recovering from COVID-19 were also infected by this fungus Aspergillus, and the disease that was caused by this fungus was Aspergillosis. A powerful toxin called aflatoxin is produced by another fungus belonging to the same genus, Aspergillus flavus. It's a genus with about 339 species. The genus Aspergillus was first noticed and documented by Antonio Micheli in 1729. When he observed Aspergillus and microscope, the conidiophores looked like holy water sprinkler, and thus he named the genus as Aspergillus. Aspergillium means holy water sprinkler. This is Antonio Micheli, the founder of scientific mycology. Aspergillus has many species. As I mentioned, there are about 339 species. But some of these species are very well known. For example, Aspergillus niger, which is very common in all food products. Aspergillus flavus, which is known for aflatoxins. In addition to that, we have Aspergillus fumigatus, which causes a number of diseases. In fact, 90 to 95% of infections in man and animals causing a disease called aspergillosis, a lung disease, is caused by Aspergillus fumigatus. Aspergillus flavus is the one which produces aflatoxins, which causes a disease called aflatoxicosis. Aspergillosis, the lung disease, which was very infamous during COVID-19 peak, and many people who were recovering from COVID-19 were infected by Aspergillus, and the disease they suffered from was aspergillosis, a lung infection. This is how the description of the aspergillosis looks like. Species of Aspergillus are important medically and commercially. Some species can cause infections in man and the others in animal. More than 60 Aspergillus species are medically relevant pathogens. They cause a host of diseases like infections in ear, skin lesions, ulcers, mycotomas, etc. This is Aspergillus niger, which is commonly found in all food products. You can see the black colored conidia. These are the different crop plants. We have dried chili, spices, peanuts, sorghum, wheat, sesame, maize. In all these crop plants, the Aspergillus species, that is Aspergillus flavus, survives. Aspergillus flavus reproduces in all these crop plants. It produces the toxin aflatoxin. Aflatoxins are very dangerous. They cause a number of diseases in both animals and humans. Usually, Aspergillus reproduces by asexual methods. It produces conidia in clusters, and the conidia are produced on conidiophores in large clusters. And conidiophores will arise from sclerotia, which is formed on the host tissue. Aspergillus is a very common contaminant of most of the food grains and foodstuffs, and it survives in the number of food grains and number of foodstuffs like corns and groundnuts and things like that. In these food items, that is corns and groundnuts, the fungus survives. Both Aspergillus niger, Aspergillus flavus survives, and thus it causes severe damage to a number of cereal crops, a number of food crops. The thallus of Aspergillus is made up of hyphae. It grows rapidly. It is an aerobic fungus with septate hyphae, and it produces different colored colonies. It ranges from bluish-green to yellow to black due to the profuse production of pigmented spores, that is conidia. There are chines of conidia that are small and oval. They are produced on tips of phyalides, radially positioned over the surface of the solen tip of the aerial hyphae, that is conidiophore, which develops at right angles from the specialized hyphal food cells, which arises from the sclerotia. And the sclerotia is formed by the mycelium of Aspergillus, which is produced in the thallus. You can see the septate hyphae of Aspergillus, and the hyphae forms the sclerotia, and the sclerotia at the time of reproduction produces the erect conidiophores, and the erect conidiophores are large branch structures. They have a vesicle. At the tips of the vesicle there are medulla, and these medulla will produce phyalides, and at the tips of phyalides chines of conidia, which are spherical, are produced, and these conidia are released from the conidiophores, and they spread the infection from one plant to the other plant. You can see here the conidia arising from phyalides. These are the different types of sclerotia. On the left hand side you are seeing the sclerotia, on the right hand side you are seeing the conidiophores, at the bottom you are seeing the conidia. These are the corns infected by Aspergillus flavus, and Aspergillus flavus produces the aflatoxins. This is the classification of Aspergillus. It comes under the class Ascomycetes and family Aspergillaceae. This is the life cycle of Aspergillus. Aspergillus mainly reproduces by asexual methods, and it produces conidiophores, and these conidiophores will produce conidia. Conidia will germinate on the host plant, and they produce mycelia, and mycelia may develop into sclerotia. From sclerotia, conidiophores arise, and thus the life cycle of Aspergillus continues. Thank you very much for watching this video. Please like, share and subscribe.